And so there's some mechanisms that help protect it from injury, and that includes the eyeball is sheltered by bony sockets, and this is positioned within the orbit. The eyelids, these act as shutters to protect the eye from environmental hazards. Our eyelashes are important for trapping fine airborne debris like dust before it falls into the eye, and then also our tears, which are continuously produced by our lacrimal glands, and they help to lubricate, cleanse, and also have a bacterial side that kills off any bacteria. Well, most bacteria. So the eye is also spherical, and it has a fluid-filled structure, and it's made of 3 different layers. So we have the sclera, the choroid, and the retina. The sclera, this is the tough outer layer of connective tissue, and it forms the visible white part of the eye. And the cornea is the anterior transparent outer layer with which the rays pass through it and then project light into the interior of the eye. The choroid is the middle layer, and it has most of our blood vessels that help to nourish the retina for those neurons, and then has a specialized to form ciliary body in the iris to help stabilize the lens. And then the retina is the innermost coat under the choroid, and this is where we have our neural layer, which has our rods and our cones. So as a refresher, we have 2 different chambers within the eye. We have the anterior chamber, and then we have the posterior chamber. So the anterior chamber is a clear watery liquid that's secreted by the ciliary bodies, and it helps to provide nourishment for both the lens and the cornea. And then this will drain into a scleral venal sinus called the canal of Schlemm back into the bloodstream. And so when we have inadequate drainage, this will cause excess pressure on that cavity, and then that will cause and may lead to glaucoma due to an increased pressure then on this vitreous humor. The interior consists of this vitreous humor that is responsible for maintaining the spherical shape of the eye. So the iris is responsible for controlling the amount of light that enters the eye, and it also contains 2 sets of smooth muscle networks. We have the reticular or constrictor muscles, and then the radial or the dilator muscles. So both of which will be innervated by the parasympathetic or the sympathetic division, where when we have constriction or contraction of the circular muscles, this is due to the parasympathetic stimulation, but when we have dilation, there's contraction of the radial muscles due to sympathetic stimulation. So the iris also has pigmented epithelium, and this is what gives us our unique eye color. And it's unique for each individual and is lately geared toward the latest identification technology because it's like your own DNA fingerprint or your own fingerprint, which is unique. The pupil is actually an opening. It's not actually a structure through which the light will enter. The parasympathetic division will have a circular constriction of the muscles running in a circular fashion, and so this will be called pupillary constriction. And so that's what we see within this image here. The sympathetic stimulation will have radial dilatory muscles that run radially, and so this will allow for pupillary dilation. So here in this example, we have the sympathetic stimulation. So the eye also has some refractive properties to it too. And so when light passes through the cornea and into the anterior chamber of the eye, it then will pass through the pupil, which will change shape due to pigmented iris muscles. And this helps to allow more or less light in. So light will actually come in, bounce off, and then go back again. And so that's what's going to stimulate then the shape of the lens. And when it passes through the lens, this will change the shape to focus that image. Then that passes through the posterior chamber and then to the vitreous body, so it keeps bouncing back and forth, finally until it hits the retina, where the photoreceptors are found, and then it's absorbed by the pigmented choroid layer. So the two structures that are involved in the eye's ability to have refractive properties include the cornea and the lens. So the cornea is responsible for contributing to total refractive ability, and it remains constant because the curvature never changes. However, the lens, though, is going to adjust to change to the curvature as it needs for near or far vision. And so this is due to the fact that it's composed of layers of living cells that are normally completely clear. However, it is avascular and has a low cellular metabolism that's in anaerobic conditions. And so these are the lens is attached to muscles called ciliary bodies and also suspended from the suspensory ligaments. Which then this leads us to accommodation of the lens, which is the ability of the lens to keep an object focused on the retina at a distance between the eye and the object that moves. And so contraction of the ciliary muscles allows for the suspensory ligaments to relax and the lens to thicken and round up. And this is good for close vision. Relaxation then of the ciliary muscles pulls on that suspensory ligament and causes the lens to thin and flatten. And so this is good for distant vision. So problems that occur are with presbyopia, which is a loss of accommodation that we normally see with age. And this is due to a decreased inflexibility of the lens. And this also, with the lens, it cannot thicken to increase refraction for near objects, unfortunately. So problems with refraction can occur and can be tested by the Snell eye chart, which measures the accommodation of the eye at 20 feet. And so the condition of emmetropia, this is of normal vision, and so there would be no correction necessary because the light coming in will project right onto the retina. Hyperopia occurs when there is distant images are brought to the point of focus behind that of the retina. And so these are often due to a short eyeball, and these are corrected then by convex lenses. Myopia here is considered as nearsightedness, and this is where distant images are brought to a point of focus in front of the retina. And these are often due to elongated eyeballs, and these can be corrected though by concave lenses in eyeglasses. Then the last case of astigmatism means that there is an asymmetrical light entry within the cornea and or of the lens curvature is asymmetrical. And so then this gives us several points that are either in front of, on, or behind even the retina. And so this has to be corrected by a cylindrical lens. The projection of our external world on our retina is going to include an opposite or contralateral side. So that being said, that means that the right side of what we see is going to project onto the left side of the retina. And then the left side of what we see is going to be projected on the right side of the retina. So when we look at the differences here between the yellows and the green, so the yellow represents the left side within our visual world. And then so that information is going to be transduced onto then the right side of the retina for both the left and the right eye. And then the opposite is true then for on the right side of our external world is going to be projected onto the left side of the retina, where then that information is going to converge from both eyes into the optic chiasm and then eventually into the optic nerve. So the neural portion of the retina is what is the extension of the central nervous system. And so this is composed of three different layers of excitable cells. The first layer is the outer portion, and so This is what is made up of rods and cones. Then we have the middle layer, and so this is composed of horizontal cells, bipolar cells, and amacrine cells. And then the inner layer is composed of the ganglionic cells. The ganglion cells have their axons, which are going to converge and to form the optic nerve. There is a point where the optic nerve leaves, and this is the optic disc of the eye. And so this is also our blind spot because we cannot project an image that's detected due to lack of rods and cones and because there is a hole for that optic nerve to exit out. So the photoreceptors, which again are an example of an exteroreceptor because they're detecting stimuli from our external environment, consist of two different segments. So the first is an outer segment, and so this is what's actually detecting light, and then we have the inner segment, which has all the organelles within the cell and has the synaptic terminals that will release neurotransmitter. So one thing to notice too, the difference between cone cells and rod cells is that the outer segment is actually larger within the rod cells versus that within the cone cells, and primarily because we use our rod cells during low light, and so they need to be larger so that they can absorb as much light as possible. So the effects of light on the rods, which rods allow black and white vision in low light. And so these contain a purple pigment called rhodopsin, which absorbs green light best. And this absorption causes then rhodopsin to dissociate into retinol aldehyde into opsin. Retinol aldehyde, which is also called retinol, is derived from vitamin A, and when we have an excess amount, this is called a bleaching reaction. So when we look at this image here, what we see is that on the outer segment of this rod, we have lots of little discs that are what make up that outer segment. So within the discs, we have those molecules of rhodopsin that when they have rhodopsin in the dark, there's a conformational change. So it turns into a different isomer. So it goes from an inactive state in a cis state to then into a trans state, which is now activated for that retinol. So when a person enters a dark room after being in light, there are fewer photopigments in rods and cones. And so it might take about 20 minutes to produce the amount of pigments that are needed and to adjust to the person's eyes. So within our retinal cells, the pigment rhodopsin absorbs photons and converts it to rhodopsin and retinol. And this activates a second messenger system. So in the dark, sodium channels are always open and are partially depolarized. So this has a continuous release of inhibitory neurotransmitter and prevents from firing action potentials. However, in the light, rhodopsin changes configuration and activates enzymes that close these sodium channels. This hyperpolarizes the cones so no action potentials are formed and the inhibitory neurotransmitter release is stopped. So if we look at this image here, we have in the dark on this panel, and what happens is that here sodium is constantly entering into the cell. And so these channels are always open and diffuses into the rod cell. Then glutamate is constantly released from the unstimulated cell. So here we have glutamate is continuously released. The glutamate then from the rod cell is going to inhibit the bipolar cell from releasing their neurotransmitter. And so we don't have an action potential that's generated. Now, in the light, what happens with the rods is that there is a change in the rhodopsin, and it's to a different configuration. And so that's actually going to close those sodium channels, and so sodium can no longer come in. And so that's going to block or result in hyperpolarization. Now, glutamate here from those rods are going to decrease in the amount of neurotransmitter release. And so what that does now is that this is going to excite those bipolar cells, and that will now generate action potentials. So the mechanism by which this phototransduction occurs within the rod cells, again, is that we have rhodopsin is going to be the primary protein, and that's found on those discs within the outer segment of those rods. When we have light that stimulates these are going to close sodium channels, and then we'll have a conformational change into transducin. And then what will happen is that these photoreceptors will become hyperpolarized and then will excite then the bipolar cells to then generate action potentials. So here, when we put it all together, let us look at differences between the dark and light stimulation. And so what we find here is that with the dark, again, this is going to take place within the rods and particularly within the outer segment. So we'll have a high concentration of sodium channels that are going to be open. So sodium is going to constantly be depolarizing that membrane. So what happens then is that we'll have opening of calcium channels in the synaptic terminal, and so this is increasing the release of inhibitory neurotransmitter. Okay, so a little bit different from what we've seen with other neurons and what we've typically associated with them. But because we have an inhibitory neurotransmitter that's released, this is inhibiting then those bipolar cells from creating an action potential that will travel then to the optic nerve. And so we'll have no action potential that's generated, and so there is no propagation into that visual cortex. Now with light though, we're going to have an opposite effect. So this is going to be due to reconfiguration of different pigments. So photopigment is going to be altered into transducin, which then is going to decrease and close the amount of sodium channels and sodium that enters into the membrane. So the membrane then now is going to be hyperpolarized, and that's going to close calcium channels, which then will stop the release of the inhibitory neurotransmitter. So now that means that the bipolar cells can be activated and excited, and they can now generate action potentials that are going to travel into the optic nerve. And so now we have then that transmission of information to the brain and what we'll perceive as visual perception. Cones now, the other photoreceptor, are responsible for color vision and are less sensitive to light, but they allow for color vision and a greater visual acuity. And so there are three different types of cone, the S, the M, and the L. S for short wavelengths, which respond to blue and absorb blue light. M for medium wavelengths, which respond to and absorb green light wavelengths. And then L, long wavelengths, which respond and absorb red light. So instead of opsins, photopigments have photoopsins with retinine. Photoopsins then vary in each type of cone, and so how we perceive light will depend on how many cones are being activated and what type of cone. And so the response then is dependent on the wavelength and also the intensity of the light. So within the photopigments for photoreceptors, there's 4 different types. So for the rods, there's only one, where this provides vision for shades of gray due to rhodopsin and due to any wavelength. However, cones are going to be a little bit different, where they're going to be selective in terms of what they respond to. So the M, L, and the S type of cones. And so when we have activation of those different types, we can have then red cones, green cones, and blue cones that are going to give us our perception of light. So from that though, what we have is that color spectrum. And when we have, say, perception of yellow, what we're actually doing is activating only our green cones and then also our red cones. When we have, say, purple, what we're doing is we're activating our blue cones with our red cones. And every color in between is due to some type of mix between all of them. Comparing the difference between rods and cones, so we have more rods, where if you see here, 100 million per retina compared to cones where we only have 3 million. Rods are responsible again for all shades of gray. Cones are responsible for color vision. Rods have a higher sensitivity as compared to cones, but they have a lower acuity, so there's a trade-off there. We activate our rods during night vision. Cones are for day vision due to high amounts of light. And then rods, in order for them to have visibility at night, they'll have convergence, a lot of convergence, whereas cones for acuity, and so they'll have little convergence. And so rods are going to be distributed more within the periphery of the retina, whereas the cones are more centralized within the fovea of the eye. So we have the greatest visual acuity and light adaptation then found within here, the fovea of the eye. And this is also known as the fovea centralis within the macula luteae. So this region has other layers of the retina that are going to be pushed aside so that light will fall directly within a group of cones. And so here, this is the only place where there is a one-to-one relationship with a ganglion to a photoreceptor of a cone. Typically within the periphery then, it's 105 to 1. So this then gives us a great visual acuity and sharpness to what we see, but it only works in good light. So for how we adapt with our light, this can distinguish objects as you enter area with more light, or it has to take a lot more time in order for you to adapt, such as with low light vision. Also, we have rapid breakdown of the cone photopigments so that we're able to adapt to that high light. So how we perceive information from our retina to our brain is through two different systems. We have the geniculostriate system and the tactile system. So with the geniculostriate system, we have the axons from those ganglionic cells that are going to project onto the lateral geniculate nucleus of the thalamus. And then this will go by way of the optic chiasm. Okay, so here's that. And then that information from the lateral portion of the retina does not cross sides, but the information of the medial portion does. So the medial portion of our eyes here is what is actually going to cross over onto the opposite sides. And so the neurons from the thalamus are going to synapse on the striate cortex of the occipital lobe. And this is what we see as our what is being seen. The tactile system has about 20 to 30% of the ganglionic cells that are going to synapse on the superior colliculus here of the midbrain, which helps with coordination of eye and body movements. And so this is to help us localize where is the information coming to or from.